N bond has double bond character, the rotation of such peptide bond is strictly prohibited. However, the first covalent bond between alpha carbon and carbon and third covalent bond between alpha carbon and amide nitrogen is purely single covalent. Hence, these bonds are free to rotate. By convention, the angle of rotation between alpha carbon and amide nitrogen is called phi, and angle of rotation between alpha carbon to carbon is called psi. The phi and psi angles of each amino acid allow protein to fall in many different directions. Now, the secondary structure of proteins. The secondary structure of protein or second level of protein structure deals with the common regular folding pattern of a polypeptide or protein. These structures are mainly arise from hydrogen bonding between electronegativity oxygen atom of one peptide bond and amide nitrogen of another peptide bond, either in the same or different polypeptide chain. 4 types of secondary structure are found in proteins. Let us discuss them one by one. Number 1, alpha helical structure. Alpha helical structure is a rod-like structure. In this structure, the polypeptide containing rigid peptide bond is tightly wound around an imaginary axis drawn longitudinally in the middle of the helix, and the R groove, either the side chain of the amino acid, protrude outward from the helical backbone. Generally, the length of alpha helix is 10 to 15 amino acid residues. The repeating unit in a single turn includes 3.6 amino acid residue, and the distance between corresponding point per turn is 5.4 angstrom along the axis. Alpha helix structure is stabilized by hydrogen bonds in the main polypeptide chain. Particularly, the CO group of each amino acid N-terminant forms a hydrogen bond with the NH group of the amino acid that is situated 4th residue ahead, means towards the C-terminant in the sequence. All the CO and NH group form a hydrogen bond except those amino acids which are close to both ends of the helix. Alpha helix may be right-handed if the helical structure appears screw type or clockwise, or left-handed structure. It is counterclockwise conformation. Essentially, all alpha helix found in proteins are right-handed. The right-handed are energetically more favorable because the steric class between side chain and the polypeptide backbone is less. The alpha helix content of each protein is widely differ and may range from 0 to 100%. For example, ferritin, an iron binding protein, contains 75% of amino acid residue as alpha helix. Single alpha helix is usually less than 5 angstrom. However, 2 or more alpha helix can interlink to form a very stable structure which can have a length of 1000 angstrom or more. For example, myosin and tropomyosin in muscle protein and keratin in hair are made up from interlink of alpha helix. Now, factor affecting the stability of alpha helix structure. Stability of alpha helix structure is mainly affected by amino acid sequence in the alpha helix region. For example, the presence of long negative charge glutamic acid or positive charge lysine in the polypeptide chain will repel each other, hence affected the helix conformation. And presence of proline and glycine causes constraint in the alpha helix, where proline residue in the peptide linkage introduce a kink in a alpha helix, hence cannot form any hydrogen bond with others. Number 2, beta pleated sheet structure or second type of secondary structure of protein. In this structure, the polypeptide chain is extended in a zigzag, therefore they are relatively flat. This zigzag chain can be arranged side by side to form a structure resembling series of pleats, and R groups of adjacent amino acid protrude from zigzag structure. Two or more beta-pleated sheets are layered close together within a protein. The layered polypeptide chain may be parallel or antiparallel. In parallel, the adjacent polypeptide segments are in the same orientation, whereas in antiparallel, the adjacent polypeptide segments are in opposite orientation. In parallel arrangement, the CO group of each amino acid is form a hydrogen bond with NH group of the two amino acid residue further along the adjacent polypeptide segment. In antiparallel arrangement, NH group of each amino acid is form a hydrogen bond with the CO group of adjacent polypeptide segment. In beta sheets, many structures, typically 4 or 5, but as many as 10 or more, may organize together. Such beta sheets can purely antiparallel, purely parallel, or mix. Generally, beta-pleated sheets contain high amount of glycine and alanine residue, which has the smallest R group. For example, fibrin protein of silk and spider web are rich in glycine and alanine residue. Number third is the beta turn, or also known as the reverse turn. Beta turn structure is the connecting element which link the ends of the two adjacent segments of antiparallel beta sheets. The structure is 180-degree turn that involves 4 amino acid residue and also known as the reverse turn. In this structure, the oxygen atom of CO group of first residue form a hydrogen bond with the nitrogen atom of NH group of the fourth amino acid. However, the peptide group of central 2 residue do not participate any inter-residue hydrogen bonding. Glycine and proline residue often occur in the beta turn, where proline makes the tight turn. Beta turn structures are mostly found in near the surface of the protein, and the central 2 residue which do not participate any inter-residue can form a hydrogen bond with water. Number 4 is the loops. Loops are random coil-like irregular structure found in protein. Loops are well known for reversal of polypeptide chain and comprises of 2 to 16 amino acid residue. They are generally found to connect alpha helix and beta sheet structure. Similar to beta turn, they are found near the surface of a protein, thus participate in interaction between protein and other molecules. Now, the tertiary structure of protein, the third level of protein structure. Each protein is made up of variety of helixes, beta sheets, and non-regular regions, which are folded in a unique 3-dimensional structure. The overall 3-dimensional structure of protein is referred to as tertiary structure of proteins. This folding of protein into native 3-dimensional conformation includes not only the position of primary backbone atoms, but also includes all the interaction between side chain atoms of distant places. The interaction is just an application of solubility rule like dissolved like. The stability of tertiary structure of protein is maintained by many non-covalent interactions in their side chain, in addition to covalent peptide and disulfide bond. Non-covalent interaction include hydrogen bond. Hydrogen bond may be formed between 2 alcohol groups, for example, serine and threonine, or between an alcohol and acid, for example, serine and aspartic acid, etc. Number 2, hydrophobic bond. The hydrophobic bond may be formed between nonpolar side chains, for example, leucine and isoleucine. Number 3, van der Waals forces. The van der Waals forces may be formed between tightly packed hydrocarbon side chains. And number 4 is the electrostatic force or salt breeze. It may be formed between positive and negative charge of side chain, for example, lysine and aspartic acid. Now, let us discuss how R groups are interacted in the tertiary structure of water-soluble globular proteins. For example, myoglobin. Myoglobin is an oxygen-binding muscle protein. Myoglobin is a water-soluble complex globular protein made up from 150 amino acid residue. X-ray diffraction studies show that about 70% of main chain is folded into 8 alpha helices, and the rest of the chain forms a turn and loops between helix. It is very compact structure and almost no empty space inside. The interior of the myoglobin is made up of nonpolar residue such as leucine, valine, methionine, and phenylalanine. However, charged residue such as aspartate, glutamate, lysine, and arginine are absent. Only 2 histidine residue found to contain in the interior that play a critical role in the binding of iron and oxygen. Polypeptide chain therefore folds so that its hydrophobic side chains are buried inside and its polar charged side chains are on the surface. The secret of burying a segment of main chain in the hydrophobic environment arises from pairing all the Nis in a CO group by hydrogen bonding in an alpha helix or beta sheet, and unpaired hydrogen bonding in peptide Nis or CO group markedly preferred water environment. The hydrocarbon side chains are tightly packed and stabilized by the van der Waals forces, which requires intimate contact. But in the case of the hydrophobic protein, for example in porin, an outer membrane protein of many bacteria, the distribution of hydrophobic and hydrophilic amino acid residue are reversed in compared to water-soluble protein. Now another structure, domains. Domains are structurally independent folding units looking like a separate globular proteins, but they are all part of the same polypeptide chain connected in the primary structure. Large proteins often have 2 or more domains, and the size of the domain range from about 30 to 400 amino acid residue. For example, troponin C, a protein found in muscle, has 2 domains which are present in one polypeptide chain, and extracellular part of the CD4, the cell surface protein, which the ICIV attest, has a 4 domain. Now quaternary structure of protein, it is a 4th level of protein structure. Many proteins contain 2 or more separate polypeptide chain, and each polypeptide chain is called a subunit. The arrangement and the nature of their interaction of such 2 or more subunits in 3-dimensional conformation is known as quaternary structure of protein. The protein with only one chain have no quaternary structure. Therefore, quaternary structure of protein deals with only the proteins which have more than one polypeptide chain. Quaternary structure is stabilized by non-covalent interaction such as hydrophobic interactions, electrostatic interaction, hydrogen bonds, as well as covalent disulfide bonds. Now naming of the quaternary protein. Quaternary proteins are named as dimer for 2 subunits, trimer for 3 subunits, tetramer for 4 subunits, and so on. And if the subunits are of same kind, then prefix homo is used, and if it is different kind, then called hetero. Example, hemoglobin is made up of 4 subunits with 2 different kinds, hence known as heterotetramer, where 2 subunits of one type alpha and 2 subunits of another type beta. Now in conclusion, the primary structure of protein or first level of protein structure is the sequence of amino acid residue in a polypeptide chain. Secondary structure of proteins or second level of protein structure is a common regular folding pattern of a polypeptide chain due to the hydrogen bonding between electronegative oxygen and nitrogen atom of adjacent peptide bonds, either in the same or different polypeptide chain. Secondary structure includes alpha helical, beta sheet, turn, and loops. Tertiary structure of protein refers to the overall three-dimensional conformation of a whole polypeptide chain in its folded state. Tertiary structure is stabilized by many non-covalent bonds, viz. Hydrogen bond, hydrophobic bond, salt bridge, and covalent disulfide bond, etc. And the arrangement of two or more subunits in three-dimensional conformation is the quaternary structure of protein.
बहुत पहले से पेड़ पौधे हमारे लिए उपयोगी होने के साथ-साथ रुचि, आश्चर्य और जिज्ञासा के विषय रहे हैं। यही कारण है कि हमने वनस्पतियों को काफी नजदीक से जानने और समझने का सतत प्रयास किया है। आज जबकि हम बहुत सारे पेड़ पौधों की प्रकृति से भली भांति परिचित हैं, कुछ ऐसे भी पेड़ पौधे हैं जिनका ज्ञान हमको या तो एकदम नहीं है या बिल्कुल कम है।
वनस्पति शास्त्रियों ने पेड़ पौधों की समानताओं एवं विभिन्नताओं के आधार पर इनका वर्गीकरण किया। बेंथम और हुकर नामक विद्वान वनस्पति शास्त्रियों ने बहुत पहले इन पेड़ पौधों का वर्गीकरण करते हुए इन्हें 200 समूह में रखा। किंतु इस वर्गीकरण की प्रक्रिया के दौरान उनका एक ऐसे वृक्ष से साक्षात्कार हुआ जो उनके लिए एक समस्या बन गया। उन्होंने इस पेड़ को एक अनियमित या विद्रोही पौधा मानकर अलग समूह में रखा। ये पौधा कोई और नहीं, सहीजन ही है।
भारत के विभिन्न प्रांतों में भिन्न-भिन्न नामों से पुकारे जाने वाले सहीजन का वानस्पतिक नाम मोरिंगा ओलिफेरा या मोरिंगा टेरिगोस्पर्मा है। मोरिंगेसी कुल का सदस्य सहीजन खाली भूमि, घरों के आसपास या बगीचों में देखा जा सकता है। सहीजन एक मध्यम आकार का वृक्ष है जिसकी ऊंचाई 6 से 10 मीटर तक होती है। तना शाखित होता है। शाखाएं मुख्य स्तंभ के ऊपरी भाग से निकलती हैं। सहीजन का तना तथा शाखाएं कमजोर होती हैं, अतः इस वृक्ष पर चढ़ना खतरनाक है। छाल का रंग भूरा होता है। छाल काफी मोटी होती है तथा छोटे-छोटे टुकड़ों में निकलती रहती है। पत्ते संयुक्त त्रिपक्षी होते हैं, यानी पत्र वृंत में 3 बार विभाजन होता है। ये इस पौधे का एक विशेष गुण है। पत्ते सर्दी के मौसम में झड़ जाते हैं। पुष्पक्रम संयुक्त असीमाक्षी या पेनिकल होता है। पुष्प सफेद या हल्का पीलापन लिए होते हैं जो गुच्छों में लगते हैं। पुष्प की विशेषता यह है कि ये षष्ठभागी होता है। यही कारण है कि इसके वर्गीकरण में कठिनाई आती है। प्रौढ़ पुष्प पूर्णतः पीले हो जाते हैं ऐसी स्थिति में पूरा पुष्पक्रम पीले, सफेद तथा क्रीमी रंगों का मिला जुला आकर्षण प्रस्तुत करता है। पुष्प से मीठी सी भीनी भीनी महक आती है जो मधुमक्खियों तथा अन्य कीटों को आकर्षित करती हैं। पुष्प सितंबर माह से ही लगने शुरू हो जाते हैं। इनकी पंखुड़ियां पीछे की ओर मुड़ जाती हैं। सहजन का फल त्रिकोण फली होता है जिसकी लंबाई 50 सेंटीमीटर तक हो सकती है। फल आरंभ में वक्रीय होते हैं। फल के पकने में करीब 3 माह का समय लगता है। फल अप्रैल से जून तक पककर तैयार हो जाता है। परिपक्व फल काफी कड़ा होकर बैंड बजाने की छड़ी जैसा दिखता है, इसीलिए इसे अंग्रेजी में ड्रम स्टिक भी कहते हैं। फल के अंदर असंख्य बीज होते हैं जो पंख युक्त होते हैं, इसीलिए इसका पुराना नाम मोरिंगा टेरिगोस्पर्मा है। सहजन एक बहुउपयोगी वृक्ष है। इसकी पत्तियों में करीब 20% तक प्रोटीन एवं अन्य लवण होते हैं। पत्तियों में विटामिन ए तथा विटामिन सी होते हैं, इसलिए पत्तियों का उपयोग स्कर्वी रोग तथा त्वचा के अन्य रोगों में किया जाता है।
वैज्ञानिक शोधों से इस बात की पुष्टि हुई है कि इसकी जड़ एवं बीजों में जीवाणु प्रतिरोधी गुण होता है। इसकी बीजों का उपयोग प्रदूषित जल को जीवाणु मुक्त करने में फिटकरी के स्थान पर किया जा सकता है। सहजन के स्वरस से रानीखेत नामक पशु रोग को दूर किया जा सकता है।
सहजन की फलियों की सब्जी बहुत ही स्वादिष्ट होती है। इस सब्जी का सेवन करके कमर दर्द, पेट के विकारों को दूर किया जा सकता है। यकृत के स्वास्थ्य के लिए सहजन की सब्जी बहुत ही गुणकारी होती है। फूलों को उबालकर छाछ या दही में डालकर रायता बनाया जाता है, जो पेट के रोगों के उपचार में काफी गुणकारी होता है। सहजन के बीजों से तेल प्राप्त होता है। इसी आधार पर इसका वानस्पतिक नाम ओलिफेरा यानी तेल प्रदान करने वाला रखा गया है। इस तेल का उपयोग महीन मशीनों, विशेषकर घड़ियों की मरम्मत में किया जाता है।
इसके अलावा सहजन का गोंद कमर दर्द को दूर करने के लिए खाया जाता है। इसकी छाल दांत का दर्द दूर कर देती है, इसलिए सहजन की पतली शाखाओं का उपयोग दातून के रूप में किया जा सकता है। इसकी जड़ की उपयोगिता यकृत के रोगों को दूर करने में है। सहजन के फूल कृमिनाशक गुण लिए हुए होते हैं, इसलिए इन फूलों की सब्जी खाने से पेट के कृमि नष्ट हो जाते हैं।
सहजन के लिए गर्म हवा अनुकूल होती है। ये वृक्ष सभी प्रकार की मृदा में उग सकता है। इसे कम पानी की जरूरत पड़ती है।
सहजन, जिसे सुरजना के नाम से भी जाना जाता है, इसकी 2 उन्नत किस्में वैज्ञानिकों द्वारा अनुमोदित की गई है। इसकी पहली किस्म जो निकाली गई थी उसे सदाबहार के नाम से जानते हैं। यह किस्म साल में 2 फलन देने में सक्षम है और इस किस्म में लगाने के एक ही वर्ष बाद आप फल प्राप्त कर सकते हैं। दूसरी उन्नत किस्म जो निकाली गई है उसे PKM2 के नाम से जाना जाता है और यह किस्म भी साल में 2 फसल देती है, लेकिन दूसरी फसल के लिए इसमें सिंचाई आवश्यक होती है।
बीजों से नए पौधे तैयार किए जा सकते हैं। बीजों की अंकुरण क्षमता 60-70% होती है। इसकी शाखाओं द्वारा वर्धी प्रजनन भी हो सकता है। शाखाओं को काटकर जमीन में रोपित करने से बहुत जल्दी ही नया वृक्ष तैयार हो सकता है।
है ना कई सारी खूबियां इस साधारण से दिखने वाले पेड़ में। आप भी इन खूबियों का फायदा उठा सकते हैं सही जन का कम से कम एक पेड़ उगाकर। अन्यथा आपको बाजार से इसकी फली लाकर सब्जी के स्वाद एवं लाभ से ही संतोष करना पड़ेगा।
Welcome back. In this session, we have to discuss about mechanism of drug action, drug receptor interactions. First of all, receptor. In biochemistry and pharmacology, a receptor is a macromolecule that receives chemical signals from outside the cell. When such chemical signals bind to a receptor, they cause some form of a cellular or tissue response. Example, a change in the electrical activity of the cell. In this sense, a receptor is a protein molecule that recognizes and responds to endogenous chemical signals. Example, the acetylcholine receptor recognizes and responds to its endogenous ligand acetylcholine. However, sometimes in pharmacology, the term is also used to include other proteins that are drug targets, such as enzymes, transporters, and ion channels. Receptor proteins are embedded in the cell's plasmatic membranes facing extracellular or cell surface receptors, cytoplasmic or cytoplasmic receptors, or in the nucleus or nuclear receptors. A molecule that binds to a receptor is called a ligand and can be a peptide or short protein or another small molecule such as a neurotransmitter, hormone, pharmaceutical drug, toxin, or parts of the outside of a virus or a microbe. The endogenously designated molecule for a particular receptor is referred to as its endogenous ligand. Example, the endogenous ligand for the nicotinic acetylcholine receptor is acetylcholine, but the receptor can also be activated by nicotine and blocked by curare. Each receptor is linked to a specific cellular biochemical pathway. While numerous receptors are found in most cells, each receptor will only bind with ligands of a particular structure, much like how locks will only accept specifically shaped keys. When a ligand binds to its corresponding receptor, it activates or inhibits the receptor's associated biochemical pathway. Now, let us discuss theories of drug-receptor interaction. The first one is occupation theory. The central dogma of receptor pharmacology is that drug effect is directly proportional to number of receptors occupied. Furthermore, drug effect ceases as drug-receptor complex dissociates. Arians and Stephenson introduced the terms affinity and efficacy to describe the action of ligands bound to receptors. Affinity, ability of the drug to combine with receptor to create drug-receptor complex is called affinity. Efficacy is the ability of the drug-receptor complex to initiate a response. Now, rate theory. In contrast to the accepted occupation theory, rate theory proposes that the activation of receptors is directly proportional to the total number of encounters of the drug with its receptors per unit time. Pharmacological activity is directly proportional to the rates of dissociation and association, not number of receptors occupied. Agonist. Drug with fast association and fast dissociation is called agonist. Partial agonist, drug with intermediate association and intermediate dissociation. An antagonist is the drug with fast association and slow dissociation. Next is induced fit theory. As the drug approaches the receptor alters the conformation of its binding site to produce drug-receptor complex. spare receptors. In some receptor systems, example acetylcholine at the neuromuscular junction in smooth muscles, agonists are able to elicit maximal response at very low levels of receptor occupancy, that is less than 1%. Thus, the system has spare receptors or receptor reserve. This arrangement produces an economy of neurotransmitter production and release. The structures of receptors. The structure of receptors are diverse and can broadly be classified into the following categories. That is type 1. Type 1 ionotropic receptors. These receptors are typically the targets of fast neurotransmitters such as acetylcholine or nicotinic and GABA, and activation of these receptors results in changes in ion movement across the membrane. They have a heterostructure. Each subunit consists of the extracellular ligand binding domain and a transmembrane domain, where the transmembrane domain in turn includes 4 transmembrane alpha helixes. The ligand binding cavities are located at the interface between the subunits. Next is type 2. G-protein coupled receptors or metabotropic. This is the largest family of receptors and includes the receptors for several hormones and slow transmitters. Example, dopamine, metabotropic glutamate. They are composed of 7 transmembrane alpha helices. The loops connecting the alpha helices form extracellular and intracellular domains. The binding site for larger peptidic ligands is usually located in the extracellular domain, whereas the binding site for smaller non-peptidic ligands is often located between the 7 alpha helices and 1 extracellular loop. These receptors are coupled to different intracellular effector systems via G proteins. Next is type 3. Kinase linked and related receptors. These receptors are composed of an extracellular domain containing the ligand binding site and an intracellular domain, often with enzymatic function, linked by a single transmembrane alpha helix. Example, the insulin receptor. Type 4, which is nuclear receptors. While they are called nuclear receptors, these are actually located in the cytosol and migrate to the nucleus after binding with their ligands. They are composed of a C-terminal ligand binding region, a core DNA binding domain which is called DBD, and an N-terminal domain that contains the AF1 or activation function 1 region. The core region has 2 zinc fingers that are responsible for recognizing the DNA sequences specific to this receptor. The N-terminal interacts with other cellular transcription factors in a ligand-independent manner, and depending on these interactions, it can modify the binding or activity of the receptor. Steroid and thyroid hormone receptors are examples of such receptors. Membrane receptors may be isolated from cell membranes by complex extraction procedures using solvents, detergents, and or affinity purification. The structures and actions of receptors are often studied by biophysical methods such as X-ray crystallography, NMR, circular dichroism, and dual polarization interferometry. Computer simulations of the dynamic behavior of receptors have been used to gain understanding of their mechanism of action. Now, let's discuss about ligands. Ligands are the endogenous substances, molecules, or compounds which bind with the receptors present in the body. Example, acetylcholine, adrenaline, noradrenaline, neurotransmitters like glutamate, aspartate, and GABA. They produce various effects and interfere with the flow of ions through channels called ligand-gated channels. Their actions may resemble with natural ligand and block the natural ligand. Agonists, antagonists, and partial agonists comes under this. Agonists or receptors can be activated by either endogenous such as hormones and neurotransmitters, or exogenous such as drugs. Agonist resulting in a biological response. A physiological agonist is a substance that creates the same bodily responses but does not bind to the same receptor. An endogenous agonist for particular receptor is a compound naturally produced by the body that binds to and activates the receptor. For example, the endogenous agonist for serotonin receptors is serotonin, and the endogenous agonist for dopamine receptors is dopamine. A superagonist is a compound that is capable of producing a greater maximal response than the endogenous agonist for the target receptor, and thus has an efficacy of more than 100%. This does not necessarily mean that it is more potent than the endogenous agonist, but is rather a comparison of the maximum possible response that can be produced inside the cell following receptor binding. Full agonists bind or have affinity for and activate a receptor, producing full efficacy at that receptor. One example of a drug that acts as a full agonist is isoproterenol, which mimics the action of adrenaline at beta adrenoreceptors. Another example is morphine, which mimics the action of endorphins at mu-opioid receptors throughout the central nervous system. Partial agonists such as buspirone, aripiprazole, buprenorphine, or norclozapine also binds and activate a given receptor, but have only partial efficacy at the receptor relative to a full agonist, even at maximal receptor occupancy. Agents like buprenorphine are used to treat opiate dependence for this reason, as they produce milder effects on the opioid receptor with lower dependence and abuse potential. An inverse agonist is an agent that binds to the same receptor binding site as an agonist for that receptor and inhibits the constitutive activity of the receptor. Inverse agonists exert the opposite pharmacological effect of a receptor agonist, not merely an absence of the agonist effect as seen with antagonist. An example is the cannabinoid inverse agonist rimonabant. A co-agonist works with other co-agonists to produce the desired effect together. NMDA receptor activation requires the binding of both glutamate, glycine and D-serine co-agonists. An irreversible agonist is a type of agonist that bind permanently to a receptor through the formation of covalent bonds. A few of these have been described. A selective agonist is selective for a specific type of receptor. Example, buspirone is a selective agonist for serotonin 5-HT1A. New findings that broaden the conventional definition of pharmacology demonstrate that ligands can concurrently behave as agonist and antagonist at the same receptor depending on effector pathways or tissue type. Terms that describe this phenomenon are functional selectivity, protein agonism, or selective receptor modulators. Next is antagonist. A receptor antagonist is a type of receptor ligand or drug that blocks or dampens agonist-mediated responses rather than provoking a biological response itself upon binding to a receptor. They are sometimes called blockers. Examples include alpha blockers, beta blockers, and calcium channel blockers. In pharmacology, antagonists have affinity but no efficacy for their cognate receptors, and binding will disrupt the interaction and inhibit the function of an agonist or inverse agonist at receptors. Antagonists mediate their effects by binding to the active or orthosteric, that is right place site, or to allosteric or the other place sites on receptors, or they may interact at unique binding sites not normally involved in the biological regulation of the receptor's activity. Antagonist activity may be reversible or irreversible depending on the longevity of the antagonist receptor complex, which in turn depends on the nature of antagonist receptor binding. The majority of drug antagonists achieve their potency by competing with endogenous ligands or substrate at structurally defined binding sites on receptors. Next is partial antagonists. In pharmacology, partial agonists are drugs that bind to and activate a given receptor, but have only partial efficacy at the receptor relative to a full agonist. They may also be considered ligands which display both agonistic and antagonistic effects when both a full agonist and partial agonist are present. The partial agonists actually act as a competitive antagonist, competing with the full agonist for receptor occupancy and producing a net decrease in the receptor activation observed with the full agonist alone. Clinically, partial agonists can be used to activate receptors to give a desired submaximal response when inadequate amounts of the endogenous ligands are present, or they can reduce the overstimulation of receptors when excess amounts of the endogenous ligands are present. Some currently common drugs that have been classed as partial agonists at particular receptors include buspirone, aripiprazole, buprenorphine, and norclozapine. Examples of ligands activating peroxisome proliferated activated receptor gamma as partial agonists are honocaol and falcarindol. Next, let us go through the types. First one, competitive. Competitive antagonists, also known as surmountable antagonists, reversibly bind to receptors at the same binding site or active site as the endogenous ligand or agonist, but without activating the receptor. Agonists and antagonists compete for the same binding site on the receptor. Once bound, an antagonist will block agonist binding. The level of activity of the receptor will be determined by the relative affinity of each molecule for the site and their relative concentrations. High concentration of a competitive agonist will increase the proportion of receptors that agonist occupies. Higher concentration of the antagonist will be required to obtain the same degree of binding site occupancy. In functional assays using competitive antagonists, a parallel rightward shift of agonist dose response curves with no alteration of the maximal response is observed. The interleukin 1 receptor antagonist IL-1RA is an example of a competitive antagonist. The effects of a competitive antagonist may be overcome by increasing the concentration of agonist. Often, though not always, these antagonists possess a very similar chemical structure to that of the agonist. Non-competitive. The term non-competitive antagonism, sometimes called non-surmountable antagonist, can be used to describe two distinct phenomena. One in which the antagonist binds to the active site of the receptor and one in which the antagonist binds to an allosteric site of the receptor. While the mechanism of antagonism is different in both of these phenomena, they are both called non-competitive because the end results of each are functionally very similar. Unlike competitive antagonists, which affect the amount of agonist necessary to achieve a maximal response but do not affect the magnitude of that maximal response, non-competitive antagonists reduce the magnitude of the maximum response that can be attained by any amount of agonist. This property earns them the name non-competitive because their effects cannot be negated, no matter how much agonist is present. In functional assays of non-competitive antagonists, depression of the maximal response of agonist dose response curves, and in some cases rightward shift is produced. The rightward shift will occur as a result of a receptor reserve, also known as spare receptors, seen in pharmacodynamics. And inhibition of the agonist response will only occur when this reserve is depleted. An antagonist that binds to the active site of a receptor is said to be non-competitive if the bond between the active site and the antagonist is irreversible or nearly so. This usage of the term non-competitive may not be ideal. However, since the term irreversible competitive antagonism may also be used to describe the same phenomenon without the potential for confusion with the second meaning of non-competitive antagonism, which is discussed below. The second form of non-competitive antagonists act at an allosteric site. These antagonists bind to a distinctly separate binding site from the agonist, exerting their action to that receptor via the other binding site. They do not compete with agonists for binding at the active site. The bound antagonist may prevent conformational changes in the receptor required for receptor activation after the agonist binds. Cyclothiazide has been shown to act as a reversible non-competitive antagonist of mGlu-R1 receptor. Next is uncompetitive. Uncompetitive antagonists differ from non-competitive antagonists in that they require a receptor activation by an agonist before they can bind to a separate allosteric binding site. This type of antagonism produces a kinetic profile in which the same amount of antagonist blocks higher concentration of agonist better than lower concentration of agonist. Memantine, used in the treatment of Alzheimer's disease, is an uncompetitive antagonist of NMDA receptor. Next is silent antagonist. Silent antagonists are competitive receptor antagonists that have zero intrinsic activity for activating a receptor. They are true antagonists, so to speak. The term was created to distinguish fully inactive antagonists from weak partial agonist or inverse agonist. Partial agonist. Partial agonists are defined as drugs that at a given receptor might differ in the amplitude of the functional response that they elicit after maximal receptor occupancy. Although they are agonists, partial agonists can act as a competitive antagonist in the presence of a full agonist as it competes with the full agonist for receptor occupancy, thereby producing a net decrease in the receptor activation as compared to that observed with the full agonist alone. Clinically, their usefulness is derived from their ability to enhance deficient systems while simultaneously blocking excessive activity. Exposing a receptor to a high level of partial agonist will ensure that it has a constant weak level of activity, whether its normal agonist is present at high or low levels. In addition, it has been suggested that partial agonism prevents the adaptive regulatory mechanism that frequently develop after repeated exposure to potent full agonist or antagonist. Buprenorphine, a partial agonist of the mu opioid receptor, binds with weak morphine-like activity and is used clinically as an analgesic in pain management and as an alternative to methadone in the treatment of opioid dependence. Next is inverse agonist. An inverse agonist can have effects similar to those of an antagonist but causes a distinct set of downstream between biological responses. Constitutively, active response that exhibit intrinsic or basal activity can have inverse agonists which not only block the effects of binding agonists like a classical antagonist but also inhibit the basal activity of the receptor. Many drugs previously classified as antagonists are now beginning to be reclassified as inverse agonists because of the discovery of constitutive active receptors. Antihistamines, originally classified as antagonists of histamine H1 receptors, have been reclassified as inverse agonists. Now, let us have a quick look at what we have discussed in this unit. The whole topic discusses about the interaction of drug toward receptor. This chapter also discussed about different drug-receptor interaction like occupation theory, rate theory, and induced fit theory. Different category of inhibitors like agonist, partial agonist, antagonists were also discussed in the topic. Different structures of receptors like ionotropic, G-protein coupled, kinase link, and nuclear receptors were also discussed in the chapter. Before we attend the next session, please try to answer the following questions. First one, explain about various theories of drug-receptor interaction. Second, explain about competitive, non-competitive, and uncompetitive antagonists. Third, explain about partial and inverse agonists. Here are some books for your further reference. The Receptor Concept, Pharmacology's Big Idea by Rank HP 2006. Principles, Receptor Theory in Pharmacology by Kanakin T 2004. On the reaction of cells and of nerve endings to certain poisons, chiefly as regards the reaction of striated muscle to nicotine and to curare by J. N. Langley, 1905. What systems can and can't do by Kanakin T 2008. On the stimulation and paralysis of nerve cells and of nerve endings by Langley J. 1901. Hope the session was useful for you. See you next time. Until then, bye.
This method consists of screening new molecules, whether they are synthetic or of natural origin, on an animal model or any biological test without having in mind hypothesis about its pharmacological or therapeutic potential. The trend is to undertake in vitro rather than in vivo tests, binding assays, enzyme inhibition measurements, activity on isolated organs or cell cultures, etc. There are many types of screening methods which I am explaining. The first one is called extensive screening, which is generally applied to totally new chemical entities coming from an original effort of chemical research or from a laborious extraction from a natural source. For such molecules, the high investment in synthetic or extractive chemistry justifies an extensive pharmacological study, central nervous, cardiovascular, pulmonary, and digestive systems, antiviral, antibacterial, or chemotherapeutic properties, etc., to detect if there exists an interesting potential linked to these new structures. In summary, a limited number of molecules are studied in a thorough manner, that is, vertical screening. The extensive screening approach has often led to original molecules. More recent examples of successful systematic screening campaigns are seen with the discovery of cyclopyrrolones, example, zopiclone, as ligands for the central benzodiazepines receptor, or of Taxol as an original and potent anti-cancer drug. Then there is random screening. In this case, the therapeutic objective is fixed in advance, and contrary to the preceding case, a great number of molecules are tested, but only on a limited number of experimental models. This method has been used for the discovery of new antibiotics. Among more recent successes of this approach, one can mention the discovery of lovastatin, also called mevinolin, which was the basis of a new generation of hypocholesterolemic agents acting by inhibition of HMG coenzyme A reductase. There is another type of screening which is called high throughput screening, or HTS. The arrival of the robotics in the 1980s, along with the miniaturization of the in vitro testing methods, meant it became possible to combine the two preceding approaches. In other words, screen millions of compounds on a large number of biological targets. This strategy has delivered numerous drugs. HTS is usually applied to the displacement of radioligands and to the inhibition of enzymes. The present trend is to replace radioligand-based assays with fluorescence-based measurements. Another type is screening of synthesis intermediates. As synthesis intermediates are chemically connected to the final products, and as they often present some common groupings with them, it is unlikely that they equally share some pharmacological properties. Among drugs discovered this way, one finds the tuberculostatic semicarbazones. They were initially used in the synthesis of antibacterial sulfathiazoles. Subsequent testing of isonicotinic acid hydrazide destined for the synthesis of a particular thiosemicarbazone revealed the powerful tuberculostatic activity of the precursor that has become since then a major anti-tubercular drug, isoniazid. Another type is new leads from old drugs. Selective optimization of side activities, or the SOSA, represents an original alternative to HTS. It consists of two steps. Screen newly identified pharmacological targets of a limited set, approximately 1,000 compounds of well-known drug molecules for which bioavailability and toxicity studies have already been performed and which have proven usefulness in human therapy. By definition, in using such a library, all hits that are found are drug-like. Number 2, optimize hits by means of traditional, parallel, or combinatorial chemistry in order to increase the affinity for the new target and decrease the affinity for the other targets. The objective is to prepare analogues of the hit molecule in order to transform the observed site activity into the main effect and to reduce strongly or abolish the initial pharmacological activity. The rationale behind the SOSA approach lies in the fact that in addition to their main activity, almost all drugs used in human therapy show one or several side effects. An example is the antidepressant minoprine. In addition to reinforcing serotoninergic and dopaminergic transmission, this aminopyridazine possesses weak affinity for muscarinic M1 receptors.
Hello friends, welcome to a lecture on neuron structure and function of neuron, condition of nerve impulse in a neuron, neurotransmitters, types and functions. The main objectives of today's lecture are to have a precise discussion about neurons, to discuss structure and function of neuron, to help students understand the condition of nerve impulse in a neuron, to have precise discussion about neurotransmitters, and to understand precisely types and functions of neurotransmitters. Let us first of all discuss neurons. Neurons are cells of the nervous system that transmit signals throughout the body. Neurons can do many different things, from sensing external and internal stimuli to processing information and also directing muscle actions. Neurons are the highly specialized cells of all nervous systems. For example, central nervous system and peripheral nervous system that are tasked with transmitting signals from one location to another. These cells accomplish this role through specialized membrane-to-membrane junctions called synapses. Most neurons possess an axon, which is long protrusion from the body soma of the neuron to the synapse. Axons can extend to distant parts of the body and make thousands of synaptic contacts, such as is the case with the central nervous system neurons of the spinal cord. Axons frequently travel through the body in bundles called nerves. The synapses are termed presynaptic and postsynaptic. The presynaptic synapse will release secretory granule contents in response to the propagation of an electrochemical signal down its axon. The released substance, termed a neurotransmitter, will then most likely bind to a specific receptor on the membrane of the postsynaptic synapse, thereby propagating the initial signal to the next neuron. The human nervous system is composed of hundreds of different types of neurons. These include sensory neurons that transmit physical stimuli such as light and sound into neural signals, and motor neurons that are responsible for converting neural signals into activation of muscles or glands. Moving on, let us discuss classes of neurons. Based on their roles, the neurons found in the human nervous system can be divided into three classes: sensory neurons, motor neurons, and interneurons. Now, sensory neurons. Sensory neurons get information about what is going on inside and outside of the body and bring that information into the central nervous system so it can be processed. For instance, if we pick up a hot coal in hand, sensory neurons with endings in our fingertips would convey the information to our central nervous system, CNS, that it was really hot. Now, motor neurons. Motor neurons get information from other neurons and convey commands to our muscles, organs, and glands. For instance, if we picked up a hot coal, the motor neurons innervating the muscles in our fingers would cause our hand to let go. Now, interneurons. Interneurons, which are found only in the central nervous system, connect one neuron to another. They receive information from other neurons, either sensory neurons or interneurons, and transmit information to other neurons, either motor neurons or interneurons. For instance, if we picked up a hot coal, the signal from the sensory neurons in our fingertips would travel to interneurons in our spinal cord. Let's now discuss classification of neurons on the basis of shape. The most unique and important structures of a neuron are the long extensions that extend out from the cell body. There are two types of extensions: dendrites and axons. Dendrites are extensions of neurons that receive signals and conduct them towards the cell body. Axons are extensions of neurons that conduct signals away from the cell body to other cells. Both axons and dendrites can extend far away from the cell body, and some human axons can reach lengths of over 3 feet. But that's nothing compared to giraffes, which have axons as long as 15 feet. Neurons can be classified according to the number of processes that extend from the cell body as multipolar neurons, bipolar neurons, and unipolar neurons. Multipolar neurons are the most common type. They have several dendrites and one axon extending from the cell body. Bipolar neurons have two processes extending from the cell body, an axon and a single dendrite. This type of neuron can be found in the retina. Unipolar neurons are generally sensory, that is afferent neurons, that have a single process, which then divides into two. One of the two processes extends outward to receive sensory information from various areas of the body, while the other process relays sensory information towards the spinal cord or brain. Now let us discuss the basic functions of a neuron. Neurons have three basic functions. These are to receive signals or information, integrate incoming signals, that is to determine whether or not the information should be passed along, and communicate signals to target cells, that is other neurons or muscles or glands. These neuronal functions are reflected in the anatomy of the neuron. Now, electrical signals in neurons. All living cells have a separation of charge across the cell membrane. The separation of charge gives rise to the resting membrane potential. Neurons and muscle cells both use brief changes in this resting membrane potential to quickly send signals from one end of the cell to the other. In neurons, electrical signals called action potentials propagate from the cell body down the axon to the synaptic terminals, while stored neurotransmitter is released. Action potentials are transient, all or none change in resting membrane potential that travel along the axon at rates of 1 to 100 meters per second. Myelin, a fatty insulating material derived from the cell membrane of glial cells, covers the axon of many vertebrate neurons and speeds the conduction of action potentials. The importance of this myelin covering to normal nervous system function is made painfully obvious in individuals with demyelinating disease, in which the myelin covering of the axons is destroyed. Among these diseases is multiple sclerosis, a demyelinating disease of the central nervous system that can have devastating consequences, including visual, sensory, and motor disturbances. Although neurons share many of the features found in other cell types, they have some special characteristics. For example, neurons have a very high metabolic rate and must have a constant supply of oxygen and glucose to survive. Also, mature neurons lose the ability to divide by mitosis. Until the late 20th century, it was thought that no new neurons were produced in the adult human brain. However, there is evidence that, at least in some brain areas, new neurons are produced in adulthood. This finding suggests an exciting avenue for possible approach to treating such common neurological diseases such as Parkinson's disease and Alzheimer's disease, which are characterized by the loss of neurons in certain brain areas. Let us now discuss neurotransmitters. Neurotransmitters are endogenous substances that act as chemical messengers by transmitting signals from a neuron to a target cell across a synapse. Prior to their release into the synaptic cleft, neurotransmitters are stored in secretory vesicles called synaptic vesicles near the plasma membrane of the axon terminal. The release of the neurotransmitter occurs most often in response to the arrival of an action potential at the synapse. When released, the neurotransmitter crosses the synaptic gap and binds to specific receptors in the membrane of the postsynaptic neuron or cell. Neurotransmitters are generally classified into two main categories related to their overall activity: excitatory or inhibitory. Excitatory neurotransmitters exert excitatory effect on the neuron, thereby increasing the likelihood that the neuron will fire an action potential. Major excitatory neurotransmitters include glutamate, epinephrine, and norepinephrine. Inhibitory neurotransmitters exert inhibitory effects on the neuron, thereby decreasing the likelihood that the neuron will fire an action potential. Major inhibitory neurotransmitters include GABA, that is gamma-aminobutyric acid, glycine, and serotonin. Some neurotransmitters can exert both excitatory and inhibitory effects, depending upon the type of receptors that are present. In addition to excitation or inhibition, neurotransmitters can be broadly categorized into two groups, defined as small molecule neurotransmitters or peptide neurotransmitters. Many peptides that exhibit neurotransmitter activity also possess hormonal activity, since some cells that produce the peptide secrete it into the blood, where it then can act on distinct cells. Moving on to classification of neurotransmitters. Neurotransmitters fall into several chemical classes based on the molecular structure. The major types of neurotransmitters include acetylcholine, biogenic amines, and amino acids. The neurotransmitters can also be classified based on function, that is excitatory or inhibitory, and action, that is direct or neuromodulatory. Now let us discuss acetylcholine as a neurotransmitter. Acetylcholine, ACH, is a simple molecule synthesized from choline and acetyl coenzyme A through the action of choline acetyl transferase. Once released, acetylcholine binds to postsynaptic receptors and is degraded by acetylcholine esterase. Acetylcholine releasing neurons are also found in the central nervous system, CNS. Neurons that synthesize and release ACH are termed cholinergic neurons. When an action potential reaches the terminal button of a presynaptic neuron, a voltage-gated calcium channel is opened. The influx of calcium ions, calcium 2 positive, stimulates the exocytosis of presynaptic vesicles containing ACH, which is thereby released into the synaptic cleft. Once released, acetylcholine must be removed rapidly in order to allow repolarization to take place. This step, hydrolysis, is carried out by the enzyme acetylcholine esterase. The acetylcholine esterase, found at nerve endings, is anchored to the plasma membrane through a glycolipid. Acetylcholine receptors are ligand-gated cation channels composed of 4 different polypeptide subunits. The activation of acetylcholine receptors by the binding of acetylcholine leads to an influx of sodium, Na positive, into the cell, and the influx of potassium, K positive, resulting in a depolarization of the postsynaptic neuron and the initiation of a new action potential. Now let us discuss biogenic amines as neurotransmitters. Biogenic amines include the catecholamines such as dopamine, norepinephrine, and epinephrine, as well as indole amines such as serotonin and histamine. Dopamine and norepinephrine are synthesized from amino acid tyrosine. Serotonin is synthesized from tryptophan. Histamine is synthesized from amino acid histidine. Biogenic amines are distributed in the brain, where they play a role in emotional behavior and help in regulating the biological clock. Additionally, some motor neurons of the autonomous nervous system release catecholamines like norepinephrine. Norepinephrine, dopamine, and histamine can be excitatory or inhibitory depending on the receptor type. Addictive drugs such as cocaine and amphetamine exert their effects primarily on the dopamine system, while addictive opiates and functional analogs of the opioid peptides which regulate dopamine levels. Now we have amino acids as neurotransmitters. Glutamate and gamma-aminobutyric acid, that is GABA, are amino acid-based neurotransmitters. The most prevalent transmitter in the human brain is glutamate, which promotes excitatory effects by increasing the probability that the target cell will fire an action potential. The next most prevalent is GABA, which is inhibitory at more than 90% of the synapses that do not use glutamate. Neuropeptides such as substance P and endorphins are strings of amino acids that are important in the mediation of pain signals. Enkephalin activity increases dramatically in pregnant women in labor. Endorphins are released in the so-called runner's high. There is also evidence that neuropeptides such as gut-brain peptides are produced by non-neural tissues in the gastrointestinal tract. Next important neurotransmitter is GABA. Several amino acids have distinct excitatory or inhibitory effects upon the nervous system. The amino acid derivative gamma-aminobutyrate, also called 4-aminobutyrate, that is GABA, is a well-known inhibitor of presynaptic transmission in the central nervous system and also in the retina. The formation of GABA occurs by the decarboxylation of glutamate catalyzed by the glutamate decarboxylase, that is GAD, GAD. GAD is present in many nerve endings of the brain as well as in the beta cells of the pancreas. Neurons that secrete GABA are termed GABAergic. GABA exerts its effects by binding to two distinct receptors, GABA A and GABA B. the GABA-A receptors form a chlorine channel. The binding of GABA to GABA-A receptors increases the chlorine conductance of presynaptic neuron. The anxiolytic drugs of the benzodiazepine family exert their soothing effects by potentiating the responses of GABA-A receptor to GABA binding. The GABA-B receptors are coupled to the intracellular G protein and active by increasing conductance of an associated potassium positive channel. Next important neurotransmitter is catecholamines. The principal catecholamines are norepinephrine, epinephrine, and dopamine. These compounds are formed from phenylalanine and tyrosine. Tyrosine is produced in the liver from phenylalanine through the action of phenylalanine hydroxylase. The tyrosine is then transported to catecholamine secreting neurons where a series of reactions convert it to dopamine to norepinephrine and finally to epinephrine. Catecholamines exhibit peripheral nervous system excitatory and inhibitory effects as well as actions in the central nervous system such as respiratory stimulation and an increase in psychomotor activity. The excitatory effects are exerted upon smooth muscle cells of the vessels that supply blood to the skin and mucous membranes. Cardiac function is also subject to excitatory effects, which leads to an increase in heart rate and in the force of contraction. Inhibitory effects, by contrast, are exerted upon smooth muscle cells in the wall of the gut, the bronchial tree of the lungs, and vessels that supply blood to skeletal muscles. In addition to their effects as neurotransmitters, norepinephrine and epinephrine can influence the rate of metabolism. The catecholamines bind to two different classes of receptors, termed the A and B adrenergic receptors. The catecholamines, therefore, are also known as adrenergic neurotransmitters. Neurons that secrete them are adrenergic neurons. Norepinephrine secreting neurons are noradrenergic. The adrenergic receptors are classical serpentine receptors that couple to intracellular G proteins. Some of the norepinephrine released from presynaptic noradrenergic neurons recycled in the presynaptic neuron by a reuptake mechanism. Epinephrine and norepinephrine are catabolized to inactive compounds through the sequential actions of catecholamine O-methyltransferase, that is COMT, and monoamine oxidase, that is MAO. Compounds that inhibit the action of MAO have been shown to have beneficial effects in the treatment of clinical depression, even when the tricyclic antidepressants are ineffective. The utility of MAO inhibitors was discovered serendipitously when patients treated for tuberculosis with isoniazid showed signs of an improvement in mood. Isoniazid was subsequently found to work by inhibiting MAO. With that, we come to the end of today's lecture. Hope you have understood it well. Thank you and goodbye.
विश्व के लगभग सभी देशों में गुआवा के नाम से मशहूर ये फल भारत में अमरूद, जामफल, पेरू, पियारा, सफरी और जंबा आदि नामों से भी जाना जाता है। सीरियम गुआवा नामक वानस्पतिक नाम वाला यह पौधा मिर्टेसी कुल का सदस्य है। इस कुल के अन्य सदस्य जामुन, नीलगिरी और बॉटल ब्रश हैं। अमरूद विश्व के लगभग सभी देशों में उगाया जाता है। इसके मुख्य उत्पादक देश फिजी, हवाई, ब्राजील, अमेरिका और भारत हैं। अमेरिका का मूल निवासी होने के बावजूद अमरूद फलों के उत्पादक देशों में भारत का स्थान अग्रणी है। भारत में लगभग 50 हेक्टेयर क्षेत्रफल में इसकी खेती की जाती है, जिससे 27,000 टन से अधिक अमरूद की वार्षिक प्राप्ति होती है। भारत जैसे कृषि प्रधान देश के लिए ये आय का एक मुख्य स्रोत है। भारत में इसकी व्यावसायिक खेती सभी राज्यों में होती है। उत्तर प्रदेश, मध्य प्रदेश, बिहार, महाराष्ट्र, असम, पश्चिम बंगाल, आंध्र प्रदेश और तमिलनाडु प्रमुख अमरूद उत्पादक प्रदेश हैं। अमरूद को उष्णकटिबंधीय क्षेत्रों का सेब कहा जाता है। इसका पेड़ लगभग 6 मीटर तक ऊंचा होता है। शाखा विन्यास हड्डी के ढांचे जैसा होता है, जो इसे खास आकर्षण प्रदान करता है। फैली शाखाओं और तांबे के रंग की पतली चिकनी छाल के कारण इसे आसानी से पहचाना जा सकता है। छाल जल्दी ही गिर जाती है और पुनः नई छाल आ जाती है। पत्तियां सदाबहार होती हैं, जो हल्के हरे रंग की, रोमिल और खुरदरी होती हैं। सम्मुख क्रम में विन्यासित पत्तियां अंडाकार या लंबवत आकार की होती हैं। पत्रवृंत छोटा होता है। पत्तियां 7 से 15 सेंटीमीटर लंबी और 3 से 5 सेंटीमीटर चौड़ी होती हैं। इन पत्तियों को मसलने पर भीनी भीनी महक आती है। फूल सफेद और सुगंधित होते हैं। ये पत्तियों के कक्ष में एकल या समूह में उगते हैं। फूल का व्यास लगभग 2.5 सेंटीमीटर होता है और इसमें 5 सफेद दल पाए जाते हैं। ये दल बाद में सफेद पुंकेसरों से ढक जाते हैं। इन पुंकेसरों की संख्या लगभग 250 होती है। फूल लगने के लगभग 3-4 माह बाद फल पकने शुरू हो जाते हैं। फल गोल, अंडाकार तथा नाशपाती के आकार के होते हैं। 5 से 10 सेंटीमीटर लंबे इन फलों की ऊपरी सतह पतली और हरे पीले रंग की होती है। मध्य सतह मांसल, दानेदार, रसदार और आम्लिक होती है। एक फल में 100 से 500 तक बीज पाए जाते हैं। कुछ फलों के बीज सख्त और कुछ के मुलायम होते हैं। बीजों में 14% एरोमेटिक ऑयल, 15% प्रोटीन और 13% स्टार्च होता है। फल सख्त, मीठे और पकने पर सुगंधित खुशबू बिखेरने वाले होते हैं। यह खुशबू फलों में पाए जाने वाले कार्बोनिल कंपाउंड के कारण आती है। फल स्वाद के साथ साथ भरपूर पोषकता भी देते हैं। इन फलों में विटामिन सी पर्याप्त मात्रा में पाया जाता है। कच्चे यानी हरे फलों में विटामिन सी की मात्रा सबसे अधिक होती है। एक 100 ग्राम के कच्चे फल में लगभग 350 से 400 मिलीग्राम विटामिन सी मिलता है, जबकि इसी मात्रा के पके फल में मात्र 50 से 100 मिलीग्राम विटामिन सी रह जाता है। अमरूद में पाए जाने वाले तत्वों में नमी 81.7%, वसा 0.3%, प्रोटीन 0.9%, रेशा 5.2% और कार्बोहाइड्रेट 10% है। इसके अलावा इसमें अन्य खनिज पदार्थ विटामिन ए और पेक्टिन पर्याप्त मात्रा में पाए जाते हैं।
विभिन्न रूप, रंग एवं आकार वाले इन फलों की विश्व में लगभग 50 किस्में उपलब्ध हैं, जिनमें से भारतीय किस्में 35 हैं। इनमें हफसी, करेला, चित्तीदार, नागपुर बीज रहित, सेबी आदि प्रमुख किस्में हैं। आजकल अमरूद बीज रहित भी उगाया जाने लगा है, लेकिन इससे फलों की संख्या एवं गुणवत्ता दोनों में गिरावट आती है। आज भारत में आम और केले की खेती की तरह अमरूद की खेती भी काफी फल फूल रही है। इसे सूखे एवं नम दोनों प्रदेशों में उगाया जा सकता है। अमरूद का प्रवर्धन बीज एवं वर्धी प्रजनन विधियों से किया जाता है।
अमरूद के बीजों की अंकुरण क्षमता कई महीनों तक बनी रहती है, इसलिए इसके बीजों द्वारा खेती का प्रचलन अधिक है। बीज गर्मी के दिनों में बोए जाते हैं और 3 से 5 सप्ताह में अंकुरित हो जाते हैं। 5 से 7 सेंटीमीटर ऊंचे हो जाने पर इन पौधों को खेतों में रोप दिया जाता है। इसके अलावा इसके पौधों को एयर लेयरिंग यानी गूटी और स्टूलिंग द्वारा भी उगाया जाता है। अमरूद का पौधा तेजी से बढ़ता है और 2-4 वर्षों बाद ही फल देने लगता है। 5-6 वर्ष बाद एक पेड़ पर लगभग 200 से 400 फल लगते हैं। पाले के प्रति अति संवेदनशील होते हुए भी अधिक ठंड वाले प्रदेशों में इसकी जबरदस्त फसल मिलती है। अमरूद का पेड़ साल में 2 बार फल देता है। वर्षा ऋतु वाले फल निम्न कोटि के स्वादहीन और पनीले होते हैं, जबकि जाड़ों में पकने वाले फल श्रेष्ठता की दृष्टि से सर्वोत्तम होते हैं। फल कच्चे हरे रंग के होते हैं। पकने पर इन फलों का रंग गहरे हरे से हरा पीला हो जाता है। ये फल तोड़ने का सबसे उपयुक्त समय है। फलों को तोड़कर विभिन्न श्रेणियों में छांटकर उन्हें टोकरियों में पैक कर दिया जाता है। अमरूद अतिशीघ्र सड़ने वाला फल है, इसलिए इसे बाजार में पहुंचने के बाद जल्दी बेच देना चाहिए। अमरूद के फल का अपना एक अलग ही रूप और आकर्षण है। ये कोई विशिष्ट या महंगा फल नहीं है, बल्कि ये आम जनता का फल है। आजकल ये फल 5 से 10 रुपए प्रति किलो के हिसाब से बाजार में मिल जाता है। एक निम्न आय वाला व्यक्ति भी इसे खरीद कर खा सकता है। इस फल की विशेषता है कि इसे कहीं भी कभी भी खाया जा सकता है। इसको छीलने और खाने के लिए किसी प्रकार का कोई विशेष प्रयत्न नहीं करना पड़ता। इसके फलों के मीठे स्वाद का आकर्षण बच्चों में विशेष रूप से देखा जा सकता है। हाँ, पेड़ से अमरूद तोड़कर खाने का आनंद ही कुछ और है। अमरूद के फल में अम्ल, शर्करा और पेक्टिन की मात्रा का बहुत अच्छा संतुलन है, इसलिए इसका उपयोग अनेक खाद्यों के रूप में किया जाता है। इसके फल को कच्चा व सलाद के रूप में खाया जाता है। इसके बीज और गूदे में म्यूसिलेज पाया जाता है।
Hello dear students, today we are presenting a special program on Peziza, important features and life cycle. The objectives of today's lecture are taxonomic description of Peziza, structure of mycelium of Peziza, reproduction in Peziza, both asexual and sexual, life cycle of Peziza, economic importance of Peziza. Peziza, with over 100 species, belongs to family Pezizaceae, order Pezizales, class Ascomycetes, and division A, Mastigomycota. Division A, Mastigomycota, is characterized by having absorptive type of nutrition and absence of motile spores in its member species. Dear students, fungi in class Ascomycetes have septate hyphae and bear specialized spores called ascospores, produced endogenously in specialized sporangium, known as ascus. Ainsworth, the famous botanist, raised the class Ascomycetes to the subdivision category as Ascomycotina, and subsequently recognized 6 classes within it, with Discomycetes as one of them. Discomycetes containing the genus Peziza constitutes the group of fungi having apothecium as ascocarp, that is, the fruiting body. The apothecial ascocarps produced have an exposed hymenium at maturity. Pezizales is a large order that contains the species commonly called operculate Discomycetes, as well as derived hypogeous forms that have evanescent asci with ascospores spread by mycophagy. Family Pezizaceae is characterized by having fleshy or brittle, sessile or feebly stalked and usually cup or disc-shaped apothecia. Peziza is the largest genus in the order Pezizales. It includes the fungal species, whereas asci line is the inner or upper surface of a cup-shaped fruiting body. The students, the ascus opens at maturity and discharges its spores into the air. Most members of this genus are difficult to identify as they look outwardly very similar. So identification and verification of microscopic features is usually required for identification. The spore ornamentation, which is visible when stained with cotton blue, is used in the identification of species. In common with other Ascomycetes, the upper surface of the fungus has a layer of cylindrical spore-producing cells called asci, from which the ascospores are forcibly discharged. Like a mushroom, Peziza produces above-ground heterokaryotic reproductive structures that come from an underground mycelium. One way that Peziza differs from mushrooms is that it produces spores on top of its cup, not underneath like a mushroom. Like species delimitations, intergeneric and infrageneric relationships of Peziza are debatable. Recent molecular analysis of rDNA and many other genes suggest that the genus is paraphyletic. Preliminary analysis based on nuclear large subunit rDNA sequences from a broad sampling of taxa within Peziza, as well as from related genera, suggests that all other genera in the Pezizaceae are nested within Peziza. Some species of Peziza appear to be more closely related to the species in other genera within the family than they are to some other species within genus Peziza. This infers that either more species would need to be drawn into Peziza, or Peziza will need to be broken up into a number of genera. The morphologically deviating species Peziza girardi appears to be the basal lineage in the Pezizaceae. The lineages broadly correlate with spore morphology and pigmentation. Dear students, morphological characters previously used to delimit species within the Peziza species complex, such as color variation of the apothecia, presence or absence of a stipe, stratified or non-stratified medullary excipal or thickness of the excipular layers, cell types in the outermost excipal, and monoliform versus filiform paraphysis are not correlated with the subgroup supported by ITS analysis and appear to be plastic. Further, the substrate and habitat have been much emphasized in the taxonomy of Peziza, but the populations on a diverse array of substrates may be closely related or indeed conspecific. Still, students, certain morphological characters are used to distinguish different species. For example, Peziza praetervisa is recognized by its flattened purple cup-like fruiting bodies. Dear students, a combination of characters makes Peziza vesiculosa relatively easy to identify. Most obvious is its preference for fruiting in clusters on horse dung or decayed straw. Peziza vesiculosa is additionally distinguished by an urn-shaped cup with a margin incurved even at maturity, and a wrinkled yellowish-brown hymeneal inner surface and a pale outer surface with minute pustules. Fruiting body in Peziza vesiculosa is 2 to 5 centimeters broad, sessile, often contorted when clustered. Margin is incurved, remaining so with age, at times erodes or cracks with age. The hymenium or the inner surface is light brown, pale yellow-brown to medium brown in color, frequently convoluted or wrinkled. The outer surface of hymenium is granulose to furraceous, sometimes indistinctly so when weathered, tan, pale buff to nearly white. Peziza domiciliana is a brownish cup fungus, while as Peziza syria is a yellow-gray to beige fungus, internally less than 5 centimeters across with a granular or brittle flesh. The spores in Peziza syria are smooth or elliptical, white-colored or creamy or yellowish. The cup exterior is white in color. Its upper surface at maturity is usually somewhat wrinkled near the center, a whitish and minutely fuzzy undersurface, round, cup-like shape when young, and a flattened, irregular shape when mature. Peziza ripanda can be identified by its growth on rotten wood or wood chips. Its brown upper surface at maturity that is usually somewhat wrinkled near the center, a whitish and minutely fuzzy undersurface, a round, cup-like shape when young, and a flattened, irregular shape when mature. Elliptical spores that lack oil droplets. Peziza depressa has a fruiting body about 20 to 40 millimeters, irregularly cup to saucer shaped. The fruiting body has short stipe and its inner surface is smooth, purple to chestnut brown. The outer surface is almost smooth, brighter than the inner surface. The fruiting body exuding a watery juice when cut. Peziza domiciliana is a brownish cup fungus. It is often found to grow through cracks in concrete. It is also found in sandstone and in crumbling building remains, as well as in carpets, plasters, shower stalls, and so on. It is not only the species that could conceivably grow in such settings, but it is the species that is usually found indoors or in concrete. Also, dear students, the closely related Peziza praetervisa is also violet-colored and prefers growing on burned ground. In general, Peziza praetervisa is more purple rather than violet-colored, like Peziza violacea. However, fruiting body color can vary depending on humidity and other factors, so they are more reliably distinguished microscopically. Peziza praetervisa has a rough, not so smooth spores with 2 polar oil drops. Fruiting bodies in Peziza praetervisa resemble flattened cups with a diameter of up to 3 centimeters. The inner spore-bearing surface, the hymenium, is purple-brown but lightens to brown with age. Also, one important thing that you have to remember is that the exterior surface is pale purple and scurfy. This species is sessile and does not have a stipe. The flesh is thin and mauve-colored. Spores in Peziza praetervisa are elliptical, hyaline, with small, fine warts on the surface and have dimensions of 12 to 14 by 7 to 8 micrometers. They are bigutulate, containing 2 oil drops at either end of the spores. Dear friends, the asci are 250 to 300 by 10 to 12 micrometers. Peziza bedioconfusa has cups of 3 to 10 centimeters across, deeply cup-shaped, becoming irregular. Inner surface is dull reddish-brown. The outer surface is similar but roughened or unpolished. There is no stipe in this species. The asci are about 140 by 12 micrometer in size. The spores are ellipsoid, finely watered, containing 2 oil drops of 17 to 21 by 8 to 10 micrometer size. The mycelium, dear students, is frequently perennial and consists of a dense network of branched septate hyphae. The cells are uninucleate, that's containing only 1 nucleus. The hyphae ramify within the substratum and form a complex system which extracts nutrients from the substratum. The apothecia are aerial extensions of the subterranean mycelial network. The mycelium is frequently perennial and consists of a dense network of branched septate hyphae. The cells are uninucleate. The hyphae ramify within the substratum and form a complex system which extracts nutrients from the substratum. The apothecia are aerial extensions of the subterranean mycelial network. They are semi-immersed in Peziza ammophila, while in certain species they are flattened. The size of apothecium varies from 1 to several centimeters. Similarly, they vary in color. For example, they are bright orange in Peziza aurantia. The exposed hymenium, which lines the interior of the apothecium, consists of elongated asci and paraphyses. Primary wall in ascospores is devoid of chitin and is composed of alpha-1,3-glucans. Barium permanganate treatment does not indicate appreciable amounts of protein and lipids in it. Ultrastructural data confirm light microscopic studies that show an expansion of the investing membrane within which further wall deposition occurs. One more important thing to remember is that the coalescing of cytoplasmic vesicles with the investing membrane creates a large vacuole, the perisporic sac, around the spore. Simultaneous with the formation of the perisporic sac, osmiophilic lumps are deposited on the primary spore wall. Dear students, the osmiophilic lumps increase, coalesce, and form the initial epispore. The epispore consists of zones of bands of varying electron opacity whose staining properties indicate complex carbohydrates. Dear students, the further deposition of material from the perisporic area results in the formation of spore ornaments made up of fibrillar strands. The staining reactions indicate that spore ornaments consist of a matrix of lipids, proteins, glycoproteins, and chitin. One more important thing to remember is that the ascospores are released from the apothecium generally in the moist environmental conditions. On germination, the ascospores give rise to a germ tube which develops into a new mycelium. Well, the asexual reproduction is absent in most of the species of Pezisa, but a few species produce conidia and chlamydospores. At the time of formation of conidia, some hyphae grow upwards and act as conidiophores. The conidiophores, students, you must remember, are long, cylindrical, erect, and septate. The tip of the conidiophores swells to form a vesicle. Conidia are formed on the surface of the vesicle. They are hyaline, elliptical, and thin-walled. They germinate on the suitable substratum and produce new mycelia. Chlamydospores are thick-walled resting cells formed intercalary on the hyphae either singly or in chains. In favorable conditions, they germinate and produce germ tubes which eventually grow into new mycelia. Definite sex organs, that is antheridium and ascogonium, are not formed in Pezisa. Sexual reproduction takes place by somatogamous copulation. Some hyphal branches in the mass of hyphae bear dense protoplasm, and the two adjacent cells of these hyphae fuse to form a dikaryotic cell. However, in certain cases, one must remember that the autogamous pairing takes place. Cell division in the dikaryotic cell occurs in such a way that the resulting cells are dikaryotic and form an ascogenous hypha. The dikaryotic hyphae grow together to form a reproductive structure called an ascocarp. The two nuclei in the terminal cell fuse and become a diploid nucleus. Also, the ascus then elongates, and the diploid nucleus by meiosis forms 4 haploid nuclei. Each haploid nucleus divides again by mitosis, resulting in a total of 8 haploid nuclei, which metamorphose into haploid ascospores. On their germination, they form the primary mycelia, which can produce microscopic hyphal mass. It represents the haploid phase of the fungus. Thus, in the life cycle of Pezisa, there is a long haploid phase followed by a short dikaryotic phase and a transitory diploid phase. The mature apothecium is a cup-shaped, sessile or subsessile structure 1 to 10 centimeters in diameter. They may be whitish yellow, as in Pezisa aurantia, or bright red or gray in color, for example, in Pezisa vesiculosus. In Pezisa arverensis, they are solitary, sessile, cupulate, extremely whitish, and nearly smooth. Their margins are slightly wavy, 3 to 10 centimeters in diameter, and often irregular in outline, but eventually spreading. Dear students, you must remember that the hymenium is concave, even wrinkled or furrowed, glabrous, light brown to chestnut brown in Pezisa arverensis. The exterior surface is white to ash gray, finely tomentose. The asci are arranged in parallel rows within the apothecium. In apothecia receiving light from above, the asci at the base of the cup are straight, while those on the sides are curved towards the light. Importantly, this orientation of the asci ensures smooth upward discharge of ascospores. The asci have a distinct amyloid ring at their apex, which stains blue with iodine. Ascospores are large, elliptical, and are discharged violently and in one lot. On the basis of ascospore characteristics, the species of Pezisa fall into 2 series. 1. Those producing smooth-walled spores without oil drops, and those producing spores ornamented by warts or ridges and usually containing 2 large oil drops. Dear students, some species of the second series yield a colored fluid from the flesh of their ascoscarp. The ascospores on germination at the suitable substratum give rise to a haploid mycelium of uninucleate cells. Also, a longitudinal section of the apothecium shows the following regions. Number 1, hymenium. It is the innermost region of the apothecium and contains asci, ascospores, and paraphyses. Paraphyses are sterile cells, often with swollen tips, and are at high turgor pressure and are placed at right angle to the surface of the apothecium. Tips of the paraphyses are very tightly together at the surface of the hymenium and create a barrier, the epithelium. The exposed hymenium, which lines the interior of the cup, consists of elongated asci, exhibiting various stages of development, and paraphyses. Both the asci and paraphyses, you must remember, are phototropic, and their tips are always bent towards incident light. To disperse spores, asci push between the paraphyses from below, shoot off their spores, and then collapse. Number 2 is the subhymenium. The middle region of apothecium is known as subhymenium or hypothecium. It's composed of thin-walled and light-colored hyphae which run parallel to the surface of the hymenium. The asci and the paraphyses arise from these hyphae. Number 3 is the exitulum. It is the outermost region of the apothecium, composed of sterile hyphae. Now, moving on to the economic importance of Pezisa. In general, the species of Pezisa are with no apparent use, and there is no concrete evidence about their edibility. However, the species found growing on soil have a tendency to increase soil fertility. The species growing on wood logs lead to deterioration of quality of timber. The species growing indoors are potential pollutants. A unique hemaggulating lectin has been isolated from Pezisa arverensis. This lectin determined to be a single-chain protein and is of biotechnological importance. That is all we had for you in this edition of the classes of botany. Hope you enjoyed the lecture.
These news items are just the tip of the iceberg. World statistics show a more grave situation about children's emotional health. It reveals that children worldwide are becoming more impulsive, anger-prone, lonely, and aggressive. They have more behavioral problems, psychosomatic problems, eating disorders, etc. Increasingly, children are getting depressed at a younger age, even at 9 to 10 years. There are many reasons why this is so. Pressure of studies, parents' expectations, uncertainty about the future.
With the recent spate of suicides among children of age group of 10 to 15 in last almost 1 or 2 months has almost awakened all the people, all the adults, and we are thinking why children are committing such heinous acts and such why this is happening in children of such a younger, tender age. But last 1 decade, we are seeing all over the world what is devastating is the depression in children, and children are getting more and more depressed, and depression is as good as becoming a kind of common cold of mental illness.
Can we, as individuals, change the situation? Maybe not, but we can certainly do something which will help our children, which will enable them to cope better with challenges, which this fast-changing world keeps throwing their way. How? Well, it was Dr. Daniel Goleman, a well-known psychologist, who underlined the importance of emotions and the need to develop emotional intelligence in children. But much before him, the famous Greek philosopher Plato had emphasized the role emotions play in human life. He said, all learning has an emotional base. We have all heard about IQ, and most of us believe that intelligence is what is reflected in marks or grades in examinations. But Dr. Goleman pointed out that EQ can matter more than IQ.
There is a scientific definition of emotional intelligence which has been given by Dr. Peter Salovey and Dr. John Mayer way back in 1990. And according to that definition, it is the set of abilities of any child or any person to perceive emotions, to understand emotions, to effectively use emotions and the content in that in the thinking process, and to reflectively regulate emotions so as to promote intellectual as well as academic growth. So this is a scientific definition, but in short, what it means for us, especially in the field of child education, is how the child can be made smart about his or her emotions as well as about others' emotions.
Emotions are an integral part of our life. In our daily life, we experience a number of different emotions. Even a child is no exception. Emotions are of 2 types, positive and negative. Love and joy are positive emotions, whereas anger, sadness, fear, anxiety are all negative emotions. Each of these emotions has many shades, and we may experience hundreds of different shades throughout the day. Emotions are popularly considered to be a matter of the heart. It is now established that the brain is the source of both thoughts and emotions.
We have 2 kinds of brain, which is one is thinking brain, which is the outermost part of the brain, which is called as neocortex, and there is inside is the emotional brain, which is mainly called as limbic system, consisting of thalamus, hippocampus, and amygdala. So there are various connections, neural circuits between these 2 parts of the brain, that is thinking brain and emotional brain.
Emotional intelligence is only partly genetic, and most of it can be learned and developed. Developing emotional literacy is the first step which leads to enhancement of emotional intelligence.
Emotional literacy skills, which are important, they are in the form of abilities and skills which can be developed in children. For example, if the child is able to understand, recognize, can notice different feelings and emotions in himself as well as others, and can label those feelings, give words to their feelings, this requires definite feeling vocabulary or emotional vocabulary for the child. And for expressing all these emotions in a constructive way, the child requires to develop the language of emotions. All these skills come under a skill of emotional literacy, which is the fundamental basic skill of emotional intelligence. If the child has these skills, then child is able to better verbalize his emotions as well as can manage emotions later on if he or she is aware of what exactly he or she is feeling at any point of time. So this is the basic skill. Then comes how the child can manage different emotions, can deal with different emotions like anger, fear, disappointment, frustration, all the negative as well as positive emotions which come daily in our lives. How the children can cope up with that, can deal with those emotions is another skill which is known as managing emotions and emotion management skill. Then how the child can delay gratification, something the child wants very badly, can wait for that thing for a minute, maybe for some more time, maybe an hour or maybe a week. So that is basically a skill which definitely has got long-term implication. Impulse control is another skill of emotional intelligence, where the child can he or she control his or her impulses in any situation. Many times we see children where they are more impulsive, where this skill is not so much developed. Then another skill of emotional intelligence is whether the child can motivate himself or other when the times are tough, and this is called self-motivation skill. Another skill which is very important in child's future is optimism skill, where can the child in given situation has optimistic thinking style and can hope for better and can go through rough passages in life also. There is another skill which is known as empathy, which is also a very important skill of emotional intelligence, where whether the child can understand, notice and recognize emotions in others, can he or she actually feel other person and kind of empathize and communicate empathetically with the other person. This skill is very important from long-term point of view, because when the child grows into adulthood, when he has to mix with people, if he or she can understand empathetically and have empathy, they can really communicate well and can have a good social networking later on. So all these skills together will form what is called as emotional intelligence as such.
At our center, we give a lot of focus, lot of time where the children are able to express themselves. Through the modules, it's very nice to see children open up. We have children who come in who are very shy, who take time to adjust in a group, and the unfolding that happens and the sort of development in confidence that they gain through the games, through the activities is amazing. The transformation that happens over the period of 3 months has to be seen to be believed. And for the last 4 years, we have been conducting classes for children in this pattern. Clap for him. Very, very good. Very nice. Yes, अजून कोण काय सांगणार आहे इमोशन्स बद्दल?
भावना म्हणजे आपल्याला जेव्हा दुःख होतं किंवा मग आपल्याला आनंद होतो तेव्हा आपल्या तोंडातनं जे उद्गार निघतात त्याला आपण भावना म्हणतो.
जेव्हा मिळालं जे मनाला हवं असतं ते मिळालं तर
Trilobites, meaning three lobes, are a famous group of extinct marine arthropods belonging to class Trilobita. They evolved during early Cambrian period some 526 million years ago and flourished throughout the lower Paleozoic era and almost declined to extinction during the Devonian. The trilobites were among the most successful of all early animals, strolling on oceans for over 270 million years. Trilobites include 10 orders, 5,000 genera and 17,000 species. Trilobites had many lifestyles. Some moved over the seabed as predators, some scavengers or filter feeders, and some swam, feeding on planktons, even probably symbiotic with sulfur bacterium. Their position in Mandibulata, that is Myrapoda plus Crustacea plus Hexapoda stem group, may be a more prudent alternative. In this program, we shall talk about phylogeny, relationship with other taxa, and morphology of trilobites, whereas the other topics as development, larval forms, fossil records, origin, divergence, and final extinction, evolutionary trends, importance will be covered in the next program on trilobites too. Phylogeny. Despite their rich fossil record, with thousands of genera found throughout the world, the taxonomy and phylogeny of trilobites have many uncertainties. Except possibly for the members of the order Phacopida, all 9 trilobite orders appeared prior to the end of the Cambrian. Relationship to other taxa. When soft part anatomy was recovered from the fossil record, the trilobites were originally allied to the Crustacea. However, this suggestion has since fallen out of favor, and their relationship with the chelicerates in a clan termed Arachnomorpha, that is Arachnata, was in vogue for some time. A position in the Mandibulata that is equal to Myrapoda plus Crustacea plus Hexapoda stem group may be a more prudent alternative. Morphology. When trilobites are found only, the exoskeleton is preserved, and that too often in an incomplete state. But in a handful of locations, identifiable soft body parts, that is legs, keels, musculature and digestive tract, and enigmatic traces of other structures, example fine details of eye structure as well as the exoskeleton, are recorded. It is from these records their morphology is alleged. Trilobites range in length from 1 mm to 72 centimeters, with a typical size range of 3 to 10 centimeters. The world's largest trilobite, Isotylus rex, was found in 1998 by Canadian scientists in the Ordovician rocks on the shores of Hudson Bay. The trilobite body is divided into 3 major distinctive sections, that is stegmata. Number 1, cephalon, that is the head. Number 2, thorax, that is the body. And number 3, pygidium, that is the tail. The exoskeleton is composed of calcite and calcium phosphate minerals in a protein lattice of chitin that covers the upper dorsal surface of the trilobite and curled round the lower edge to produce a small fringe called the doubler. As might be expected for a group of animals comprising approximately 5,000 genera, the morphology and description of trilobites ought to be complex. However, despite morphological complexity and an unclear position within higher classifications, there are a number of characters that distinguish the trilobites from other arthropods. A generally sub-elliptical, dorsal, chitinous exoskeleton divided longitudinally into 3 distinct lobes, from which the group gets its name, having a distinct relative large head shield, that is the cephalon, articulating axially with a thorax comprising articulated transverse segments, the hindmost of which are almost invariably fused to form a tail shield, that is pygidium. While describing differences between trilobite taxa, the presence, size and shape of the cephalic features are often taken into consideration. During molting, the exoskeleton generally split between the head and the thorax. Hence, many trilobite fossils are missing one or the other counterpart. In most groups, facial sutures on the cephalon help to facilitate molting. Similar to lobsters and crabs, trilobites would have physically grown between the molt stage and the hardening of the new exoskeleton. Terminologies. Cephalon. Morphology of the trilobite cephalon. The subdivisions can be further broken down into different areas used in describing trilobite cephalic morphology. 1. Preocular area. 2. Palpebral area. 3. Postocular area. 4. Posterior lateral projection. 5. Occipital ring. 6. Glabella. 7. Posterior area. 8. Lateral border. 9. Labyrinthinal area. 10. Preglabelar area. The cephalon of trilobites is highly variable with a lot of morphological complexity. The glabella forms a dome underneath which set the crop or the stomach. Generally, the exoskeleton has few distinguishing ventral features, but the cephalon often preserves muscle attachment scars and occasionally the hypostome. A small rigid plate comparable to the ventral plate in other arthropods. A toothless mouth and stomach lay upon the hypostome. Facial sutures. Facial or cephalic sutures are the natural fracture lines in the cephalon of trilobites. Their function is to assist the trilobite in shedding its old exoskeleton during ecdysis, that is molting. The types of sutures found in different species are used extensively in the taxonomy and phylogeny of trilobites. Rostrum. The rostrum or the rostral plate is a distinct part of the doubler located at the front of the cephalon. It is separated from the rest of the doubler by the rostral suture. During molting in trilobites, like paradoxides, the rostrum is used to anchor the front part of the trilobite as the cranidium separates from the labyrinthina. The opening created by the arcing of the body provides an exit for the molting trilobite. It is absent in some trilobites like Lenchostoma. Hypostome. The hypostome is the hard mouthpart of the trilobite found on the ventral side of the cephalon, typically below the glabella. Hypostome can be classified into three types based on whether they are permanently attached to the rostrum or not, and whether they are aligned to the anterior dorsal tip of the glabella. Thorax. The thorax is a series of articulated segments that lie between the cephalon and pygidium. The number of segments varies between 2 to 61, with most species in the 2 to 16 range. Each segment consists of the central axial ring and the outer pleura, which protected the limbs and gills. The pleurae are sometimes abbreviated or extended to form long spines. Trilobite fossils are often found enrolled, that is curled up, like modern pillbugs for protection. Evidence suggests enrollment helped protect against the inherent weakness of the arthropod cuticle that was exploited by Anomalocrarids, the dense predators. Some trilobites achieved a fully closed capsule, example Phacops, while others with long pleural spines, example Selenopeltis, left a cap at the sides or those with a small pygidium. Pygidium. The pygidium is formed from a number of segments and telson fused together. Segments in the pygidium are similar to the thoracic segments bearing biramus limbs, but are not articulated. Trilobites can be described based on pygidium being micropygus, that is smaller than the cephalon, subisopigus, that is sub-equal to cephalon, isopygus, that is equal to the size of a cephalon, or macropygus, that is larger than a cephalon. Prosopon, that is the surface sculpture. Trilobite exoskeletons show a variety of small-scale structures collectively called prosopon. Prosopon does not include large-scale extensions of the cuticle, example hollow pleural spines, but to finer scale features such as ribbings, domes, pustules, pittings, ridging, and perforations. The exact purpose of the prosopon is not resolved, but suggestions include structural strengthening, sensory pits or hairs, preventing predator attacks, and maintaining aeration while enrolled. In one example, alimentary ridge networks easily visible in Cambrian trilobites might have been either digestive or respiratory tubes in cephalon and the other regions. Spines. Some trilobites, order Lecida, evolved elaborate spiny forms. Examples of these specimens have been found in the Hammer-Legdat formation of Alnif in Morocco. Spectacular spined trilobites have also been found in western Russia, Oklahoma, USA, and Ontario, that is Canada. Some trilobites had horns on their heads similar to those of modern beetles. Based on the size, location, and shape of the horns, the most likely use of the horns was combat for mates, making the Asaphida family Rhaphidophorida the earliest exemplars of this behavior. Another use for these spines is protection from predators. When enrolled, trilobites could protect their softer innards. A conclusion likely to be applicable to other trilobites as well, such as in the Phacopid trilobite genus Velicerops that developed spectacular tridents. Soft body parts. Only 21 or so species are described from which soft body parts are preserved. So some features, example, the posterior anteniform cerci, preserved only in Olenodus serratus, remain difficult to assess in the wider picture. Appendages. Trilobites had a single pair of preoral antennae and otherwise undifferentiated biramus limbs. 2, 3, or 4 cephalic pairs, followed by a variable number of thorax plus pygidium pairs. Each exopodite, that is the walking leg, had 6 or 7 segments homologous to other early arthropods. Exopodites are attached to the coxa, which also bore a feather-like epipodite or gill branch, which was used for respiration and, in some species, swimming. The base of the coxa, the gnathobase, sometimes have heavy spiny adaptations that were used to tear at the tissues of prey. The last exopodite segment usually had claws or spines. Many examples of hairs on the legs suggest adaptations for feeding, as for the gnathobases, or sensory organs to help with walking. Digestive tract. The toothless mouth of trilobites was situated on the rear edge of the hypostome, that is facing backwards, in front of the legs attached to the cephalon. Mouth is linked by a small esophagus to the stomach that lay forward of the mouth, below the glabella. The intestine led backwards from there to the pygidium. Feeding limbs attached to the cephalon are thought to have fed food into the mouth, possibly slicing the food on the hypostome and or gnathobases first. Alternative lifestyles are suggested, with the cephalic legs used to disturb the sediment to make food available. A large glabella, implying a large stomach, coupled with an impendent hypostome has been used as evidence of more complex food sources, example, possibly a carnivorous lifestyle. Internal organs. While there is a direct and implied evidence for the presence and location of the mouth, stomach and digestive tract, the presence of heart, brain and liver are only implied, although present in many reconstructions with little direct geological evidence. Musculature. Although rarely preserved, long lateral muscles extended from the cephalon to midway down the pygidium, attaching to the axial rings, allowing enrollment, while separate muscles on the legs tucked them out of the way. Sensory organs. Many trilobites had complex eyes. They also had a pair of antennae. Some trilobites were blind, probably living too deep in the sea for light to reach them. As such, they became secondarily blind in this branch of trilobite evolution. Other trilobites, example Phecus rana and Urbankili erbini, had large eyes that were for use in more well-lit, predator-filled waters. Antennae. The pair of antennae suspected in most trilobites or the creatures such as happened shortly before the end Permian mass extinction event. With so many marine species in with elementary prosopon and a micropygium until the early 1900s, the Ate Indians of Ata
Stem cells are undifferentiated biological cells in multicellular organisms that can differentiate into specialized cells and can divide to produce more stem cells. A stem cell is essentially a blank cell that is capable of becoming another more differentiated cell type in the body, such as skin cell, a muscle cell, or a nerve cell. They are microscopic in size and can be used to replace or even heal damaged tissues and cells in the body. In a developing embryo, stem cells can differentiate into all the specialized cells like ectoderm, endoderm, and mesoderm, and maintain the normal turnover of regenerative organs such as blood, skin, or intestinal tissues. In adult organisms, stem cells and progenitor cells act as a repair system for the body, replenishing adult tissues. The history of research on adult stem cells began more than 60 years ago. Scientists first studied the potential of stem cells in mouse embryos over 2 decades ago. It was only in 1998 that researchers first extracted the first human embryonic stem cells that were able to be kept alive in the laboratory. The embryonic stem cells can be taken from umbilical cord blood just after birth. The autologous cells are obtained from one's own body. The embryonic cell lines and autologous embryonic stem cells generated through somatic cell nuclear transfer or dedifferentiation have also been proposed as promising candidates for future therapies. There are 3 known sources of autologous adult stem cells in humans. Bone marrow, from which stem cells are extracted by drilling into bone of femur or iliac crest. Adipose tissue, from which stem cells are extracted by liposuction. Blood, from which stem cells are extracted through apheresis and passed through a machine that extracts the stem cells and returns other portions of the blood to the donor. Stem cells differ from other kinds of cells in the body. The classical definition of a stem cell requires that it possesses 3 properties. Self-renewal. The stem cells may replicate or proliferate many times. That is, it has the ability to go through numerous cycles of cell division for long periods while maintaining the undifferentiated state. Potency. The stem cells should be potent to exhibit totipotent or pluripotent. That is, the capacity to differentiate into specialized cell types. Unspecialized. One of the fundamental properties of a stem cell is that it does not have any tissue-specific structures that allow it to perform specialized functions. These unspecialized stem cells can give rise to many different specialized cells. The process of differentiation is controlled by internal and external signals. The interaction of signals during differentiation causes the cell's DNA to acquire epigenetic marks that restrict DNA expression in the cell and can be passed on through cell division. The function of stem cell is regulated by a feedback mechanism. In mammals, there are 2 broad types of stem cells: embryonic stem cell, which are isolated from the inner cell mass of blastocysts, and adult stem cells, which are found in various tissues. The embryonic stem cells are derived from a 4 to 5 day old human embryos in the blastocyst phase of development. The embryos are usually extras that have been created in vitro fertilization clinics, where several eggs are fertilized in a test tube, but only one is implanted into a woman. or they may be donated for research purposes with informed consent of the donors. The zygote, after 4 to 6 days of fertilization, becomes a mass of cells called a blastocyst. The blastocyst consists of an inner cell mass and an outer cell mass. The outer cell mass becomes part of the placenta, and the inner cell mass differentiate to become all the structures of an adult organism. This mass is the source of embryonic stem cells called totipotent cells. When extracting embryonic stem cells, the blastocyst stage signals when to isolate stem cells by placing the inner cell mass of the blastocyst into a culture dish containing a nutrient-rich broth. In the absence of stimulation to differentiate, they begin to divide and replicate while maintaining their ability to become any cell type in the human body. Eventually, these undifferentiated cells can be stimulated to create specialized cells at various points during the process of generating embryonic stem cell lines. Scientists perform characterization to see the viability of these cells. They exist through the body after embryonic development and are found in tissue and organs including brain, bone marrow, peripheral blood, blood vessels, skeletal muscle, skin, teeth, heart, gut, liver, ovarian epithelium, and testis. They are thought to reside in a specific area of each tissue called a stem cell niche. They can differentiate into cells of same lineage. The adult stem cell can renew itself indefinitely and can differentiate to yield some or all of the major specialized cell types of the tissue or organ. The primary roles of adult stem cells in a living organism are to maintain and repair the tissue in which they are found. Stem cells may remain quiescent for long periods of time until they are activated by a normal need for more cells to maintain tissues or by disease or tissue injury. Scientists have found adult stem cells in many more tissues than expected, like in brain and heart. This finding has led researchers and clinicians to consider use of adult stem cells for transplants. If the differentiation of adult stem cells can be controlled in the laboratory, these cells may become the basis of transplantation-based therapies. Now, let us see the similarities and differences between human embryonic and adult stem cells. Both have advantage and disadvantage regarding potential use of cell-based regenerative therapies. One major difference between adult and embryonic stem cells is their different abilities in the number and type of differentiated cell types they can become. Embryonic cells can become all cell types of the body because they are pluripotent, while adult stem cells are thought to be limiting to differentiating into different cell types of their tissue of origin. The embryonic cells can be grown relatively easily in culture, while adult stem cells are rare in mature tissues, so isolating these tissues from an adult tissue is challenging. The adult stem cells and tissues derived from them are less likely to initiate rejection after transplantation by the immune system. This is because a patient's own cell could be expanded in culture, coaxed into assuming